Good morning, everybody. Welcome to the MPG Primer today. Our speaker today is Kiran Garimella. Kiran is the Director of Computational Biology in the Data Science Platform at the Broad, where he's also an Institute Scientist. Kiran has extensive experience in the development and application of cloud-based data processing methods and automated analyses for third-generation sequencing data. He currently leads the Data Sciences Platform's ofLong-Read group, which aims to transform the genomics landscape by undertaking long-read sequencing of human and infectious disease genomes. He earned his PhD in genomic medicine and statistics from the University of Oxford's Wellcome Trust Center for Human Genetics, and he also completed postdoctoral studies at the Oxford Big Data Institute. He co-chairs the NIH's All of Us Long-Read Working Group and also the NIH's Gabriella Miller Kids First Long-Read Working Group. Which are both undertaking the discovery and analysis of previously unseen variation in about 20,000 diverse individuals for population genomics and rare disease applications. And today he's going to be doing a part 2 on long-read sequencing, which I'm really excited about. I've had the chance to play with long-read data myself, and to load it into IGV and to keep scrolling and to see the read keep going is, it's amazing. So really excited. Kiran, take it away.
All right. Fantastic.
Oh, please ask questions along the way. Thank you.
All right, fantastic. Can everyone hear me? That's fantastic. In fact, talk to me after the meeting. There is, I don't know if you've joined the working group, but we have, okay.
Lost the NIH, but now they found them.
Okay. More than happy to talk about it after the meeting, but that's very exciting. All right. Okay. First of all, thank you for having me back. I think I was here in June, July, something like that. And I gave what I had intended to be a fairly comprehensive overview of long-read sequencing. There was so much to talk about that I didn't get to everything, so I'm very happy to have this opportunity to come back and share some of the things that I didn't get to speak about last time. If you didn't see the talk last time, I'm just going to leave this slide up for a second. You can go to the QR code. It'll take you to the YouTube video. But last time, I talked about, in general, how the PacBio and Oxford Nanopore platforms worked. I talked about some of the sort of basic operations in terms of alignment of reads to a reference sequence. I talked about SNP and indel and structural variant calling and copy number variant calling, and a few other topics along the way. If people did see that talk, I'm more than happy to answer questions about it here along the way. But I'm going to skip a lot of the details that I presented in that last presentation. So please, if you are new to long-read sequencing and starting to just trying to get a handle on the data, go there and you're more than welcome to reach out to me as well. I think my email address is probably somewhere on this slide. But that's what I'm here for. I'm more than happy to answer questions and get people acquainted with this data. Today, I'm going to talk about a few things. I'm going to talk about just a quick review of how the instruments work, not quite as in-depth as I went in last time. And I'm going to talk about 3 additional topics. The first is some updates to these instruments. It's only been a few months, and they're still exciting updates to both of these platforms. That's how fast this field is moving. I'll also talk about methylation calling, which I didn't get to in the last talk, and I will talk about some of the targeted sequencing capabilities of the Nanopore platform. So let's quickly recap the basics. What are we doing with long reads? What are we talking about when we talk about long reads? I think most of the audience here will probably be familiar with short read sequencing, 150 to 200 bases, paired end format. Kind of the workhorse of our field today, but with limitations. Right, there are places in the genome, particularly repeat expansions, low complexity sequence in general, or just places in the reference that are quite divergent from the sample of interest, where short reads just fail to align and fail to give you a decent picture of what variation at that locus looks like. Long read sequencing, about 2 orders of magnitude longer in read lengths, and with that comes the ability to map much more confidently across repetitive regions, across large structural variants, being used to help create new versions of reference sequences with all of the gaps closed. This is the telomere to telomere project and related the Human Pangenome Reference Consortium. We've been using them for the All of Us project to get a clearer picture of structural variation in a population genetic setting. Been using them in Gabriella Miller Kids First to get a clearer picture of potentially causal variation, namely structural variation and repeat expansions in rare disease contexts. A few years ago, the Broad had 1 of these instruments as a prototype just for some experimentation. Now it's become a full-on production device. I don't remember how many instruments are in BCL. It changes as instruments get upgraded and swapped out, but I think there are somewhere between 5 and 10 of the latest instruments, and a nanopore instrument as well. I think that makes the Broad the largest long-read sequencing center in the world at the moment, but I wouldn't be surprised if those numbers fluctuate over time. What are we doing with these structural variants? I like this example. This is from one of my colleagues, Fritz Sedlacek at Baylor College of Medicine. And in these 2 images, he depicts very clearly what the value is of long-read sequencing versus short-read sequencing. So the top track is Illumina sequencing. On the left, deletion. On the right, an insertion. On the left, you see this deletion kind of clear as day in the short read data, but what you're seeing is just an absence of sequencing data. It's actually hard to distinguish that between a true deletion versus a place that just didn't get any coverage whatsoever. Paired end reads helps with this, but you can see the reads in this IGV screenshot that are colored something other than gray indicate that the pair, the mate of this end of the paired end read is mapped somewhere that you wouldn't expect, right? So even with that paired information, it can be hard to reconstruct deletions of a certain size. Whereas the 20,000 base or so reads that you get from both PacBio platforms and nanopore platforms read through that without ambiguity, right? You know that this is not a coverage loss. You know that there is an actual deletion at this locus as evidence from the reads. Same thing with the insertion. The Illumina data, there seems to be some signal in the paired end reads, but again, there are a lot of mismapped pairs, and it's really unclear. It's actually surprisingly unclear from this example what is going on here. PacBio sequencing, Oxford nanopore sequencing identify the insertion fairly easily. So I spent my PhD and my postdoc trying desperately to reconstruct loci like this from short read sequencing. About halfway through my PhD, I got long read sequencing, and it sort of obviated the past 3 years of work, which is annoying at the time, but really exciting today when you see these problems that have been so difficult to get traction on for the past 10 or 15 years just appear in your data. It's really wonderful to see. How do the instruments work? I usually present this in the order of Illumina sequencing, PacBio sequencing, and nanopore sequencing. Just let's review very quickly how Illumina sequencing, and you'll see why I present it in this order. So Illumina sequencing, you could think of it as kind of trying to trick a polymerase enzyme into doing our dirty work here, right? You start with single-stranded DNA fragments to which you attach sequencing adapters. Complementary adapter is attached to the flow cell. As you flow in all of the fragments, They bind to places on that flow cell, perform PCR extension, bridge amplification in order to generate a local cluster of identical templates. Those are then dissociated into single-stranded fragments. Those form clusters, and one cycle at a time, you flow in nucleotides with fluorescent labels that attach to the next available occupancy spot in the single-stranded fragment. Pause, image, the superposition of fluorophores at each cluster registers a strong enough signal that a camera can read that out and call a base, and then you proceed to flush the fluorophore from the previous cycle, add the next one in. Stop and go. You are essentially sequencing this base one at a time, sequencing by synthesis. PacBio sequencing is also hijacking a polymerase in order to do its dirty work, but rather than forming these colonies of identical templates, the flow cell is designed with small wells that only house ideally one polymerase enzyme. So the correct operation of these wells is a single polymerase enzyme affixed to the bottom of the well, sequencing one read at a time. And again, we're flowing in fluorescently labeled nucleotides. Now, the key difference here between Illumina sequencing, which is using a cluster of templates in order to register a signal, versus PacBio, which is using literally one read, not a colony of reads, not a cluster of reads, is the fact that the labeled nucleotides are freely diffusing in this thermal bath, except when they are being incorporated into the single strand template. At that time, the polymerase is holding that nucleotide in a single spot for much longer than the mean diffusion time of the nucleotides. That is enough, plus the construction of the well in order to amplify light, that is enough for a movie camera to image the read in real time. So what you get is a readout that looks like the one below, where you have light intensity, the fluorescence of each nucleotide in 4 different colors, but you also have a number of kinetic readouts. You have how long did that pulse last? What is the time between the pulses? Usually referred to as pulse width and interpulse distance, right? So you're getting more information, you're reading out this information in real time, and then the instrument base caller is distilling that into nucleotide calls. To get accuracy, again, one of the ways that you get accuracy from Illumina sequencing is that you have a colony of reads that all say the same thing. So most of the fluorophores should agree with one another and register a strong signal. That signal, those colonies shrink as you do sequencing, just as you do the flush with each cycle. Some of those fragments will wash away, and so the signal attenuates over the course of the sequencing experiment. It doesn't happen for single cell sequencing, right? Or single molecule sequencing, right? There isn't anything being flushed away. So basically, from the start of the read to the end of the read, it's more or less equal quality. It's still noisy. You're reading from a movie rather than from a still image, and it can be quite difficult to know, have I seen 3 A's or 4 A's or 5 A's or a repetitive sequence motif. So there's still some error correction that needs to be applied. In order to achieve that, PacBio constructs its fragments for sequencing with a hairpin adapter on each end called a smart bell adapter. And that allows the polymerase to anneal to one of the open parts of the hairpin adapter and begin sequencing. This polymerase has strand displacement activity. So as it is synthesizing the complementary strand, it's also displacing the strand that was already there. So you get these circular readouts of a single fragment, both forward and reverse direction, usually something like 5 to 15 passes around the molecule. And with those redundant passes, those redundant passes can be combined in order to generate a high accuracy consensus read. We generally think that the corrected read is on par, maybe a little bit higher quality than Illumina sequencing today. Nanopore sequencing. Now, the interesting thing about nanopore sequencing is, unlike Illumina sequencing and PacBio sequencing, Nanopore is not using a polymerase in order to incorporate bases and just monitor the activity of the polymerase. Instead, Nanopore is an electrical readout. So a engineered pore sits in a lipid bilayer, and as fragments pass through that pore, they generate a current signal. That current is characteristic of whatever is being read, whether it's a modified or unmodified nucleotide. Can also read RNA bases without having to convert them necessarily to cDNA. So it's a much more flexible readout. The only thing that one would theoretically need to do in order to get it to read something else is to train the machine learning base caller to recognize a signal other than ACTG or U. Again, there are clever things that Nanopore does in order to take its electrical readout, which is again kind of a movie, and turn that into a high accuracy base caller. I think similar to PacBio, there are some clever hairpin structures and duplex sequencing. There are a lot of different schemes. There's also a huge amount of innovation on the base caller side. So I think Nanopore releases a new version of its base caller. I don't know, it feels like every 3 to 6 months. But there is constant improvement in the base caller. There's constant improvement in their engineered pores. The most recent innovation is essentially a dual reader head pore that basically generates 2 electrical signals per readout. So you get kind of redundant sequencing without the circularized DNA fragment. You essentially get two chances to read it as it translocates through the pore. Okay. That is a brief recap of what I talked about last time. I go into more detail in the previous talk. So again, just for a second, I'll put up the slide in case anyone wants to go to the previous link. Okay, hopefully that's enough time. Instrument upgrade. So 3 things I want to talk about today. I want to talk about some of the improvements that have happened with the instruments, methylation, and targeted sequencing. So already, like I said, I was only here a few months ago, and already my slides from that time are a little bit out of date. Just in the past couple of months, I think ASHG, PacBio announced a new addition to their lineup, the PacBio Vega. The Vega is meant to be a benchtop version of their PacBio Revio instrument. With slightly lower lower yield, but still able to generate high quality, what PacBio calls hi-fi data, this error corrected data. Nanopore has had benchtop instrument. I mean, all of Nanopore's instruments are benchtop, and I think the last time I showed the box in the back that had 24 or 48 flow cells, and the box on the right that had 5 flow cells, and some of the handheld ones, which are very fun to play with. I used to have one. I gave it away. How great is it that we're at a time when I can just buy a sequencer and put it in my pocket? But since then, they've come up with with some additional formats as well that are meant for individual labs and just benchtop use. Things that devices that support 1 or 2 flow cells, rather than needing to constantly feed some of the more production capable instruments that take 24 or 48 flow cells. Those can be hard to keep occupied for a long time. PacBio has also announced some improvements in their chemistry. In fact, I think the Broad should be getting preliminary data on this probably in the next week or 2. So this is a very recent development. A new chemistry from PacBio called Spark, which PacBio claims achieves a 33% increase in yield per smart cell. So each smart cell at the moment, each Revio smart cell is expected to generate somewhere between 24 and 30x coverage. And PacBio is releasing this new chemistry that they claim will generate somewhere between 35 and 40x coverage. I think the way they think about this is 220x genomes per smart cell. Requiring less DNA, right? Previous input requirements were around 2 micrograms. Now we're down to half a microgram. So the capability of doing more precious samples. I know a lot of my colleagues in the cancer community really worry about this number because the amount of DNA required for PacBio sequencing is substantially larger than it is for Illumina sequencing. Yes.
Like I have an intuition.
Yes, you can use the next one.
So I have an intuition of what like 30x coverage means for Illumina.
Yeah.
In your mind, do you translate that to like Illumina 30x? Does that mean something a little bit different than 30x here given accuracy or anything? I'm just curious, like if my interpretation of what 30x means from Illumina is similar.
Yeah, it's an excellent question. So the way that I think about the way that I think about 30x, the way that I think about 30x is actually what does it take to accurately call all of the variants that you expect from a truth data set. And you could do this evaluation in a number of different ways. I think the most common way to do it is titration experiment. I have some data set that was at very high coverage that I'm very confident in that was sequenced to, let's say, 40x, 60x, 80x, 100x, something where I think that I've genuinely discovered more or less everything that there is to discover. That is not an easy experiment to do in the first place, and we're getting better at it with the release of a Human Pangenome Reference Consortium data and things like that. But then the question is, all right, I'm going to sequence to a lower coverage. How much of that data did I recover? Now there are two things to consider there. One, the capabilities of the software will vary. So for example, structural variant callers may not be confident just seeing 1 or 2 reads supporting a large structural variant. They may require 3 or 5 reads. You may also require more data for genotyping. So you can do this as I'm just going to downsample my data to 30x, 25x, 20x, 15x, et cetera, et cetera. Now, the second thing to know is that long read coverage is much more even than short read coverage. And a lot of the discussion around should we have 20x PacBio sequencing and nanopore sequencing versus 30x, I think it falls into this consideration where you require higher coverage in Illumina sequencing in order to shore up the parts of the genome that tend to get lower coverage. That happens less with PacBio and nanopore sequencing. And so the recommendation that we can hit, I mean, you can see from, so I'm showing a plot from PacBio, but there are actually a number of different plots that look pretty much exactly like this. My group has done work very similar to this that is in broad agreement. But the idea is that we know we're missing quite a few things at 10x, we're missing less at 20x, and the gap between 20x and 30x is not so large. Now, I want to be clear, that is not me suggesting that all experiments on long-read instruments should be done at 20x, right? This will obviously depend on the application. I'm more than happy with the 15x that we're generating for the All of Us project, but the goal of the All of Us project is not necessarily comprehensive structural variant detection down to singletons in every single sample. It's more to construct a resource of structural variants that could be utilized by other projects. And seeing more samples is better than seeing every single variant in one sample. That's not necessarily true for rare disease projects. One of my new colleagues is looking at somatic variation in samples that would call for higher coverage. So, but that's where this difference between the recommendation for 30x at Illumina and 20x for long-read platforms is coming from. A bit of a long-winded answer, but I hope that answers your question. Where was I? So improvements in PacBio's chemistry, also improvements in, already this slide is out of date, but already additional improvements in with nanopore sequencing as well. Again, like I said, the two things that Nanopore tends to innovate on are the construction of their pore, which they label with an R and some numerical identifier. I think we're up to R 10.4.1 for is their latest pore. And innovations in base calling. So they're achieving somewhere between 100 and 120 gigabases per read, per flow cell, on par with what we're seeing from PacBio instruments. PacBio data is at the moment higher quality. Nanopore impressively seems to keep catching up. So I'm very happy to have these two companies competing with each other to drive their instruments to greater and greater heights for our use. I won't go into the duplex sequencing, but basically what I was saying before, there are a lot of different library schemes that allow you to correct some of the errors in the sequencing data. This is what I wanted to get to, methylation calling. So last time, and even in this talk, I've talked about base calling for PacBio data and Nanopore data, basically that either fluorescence readout over time or the electrical current readout over time, as allowing one to call ACs, Gs, and Ts directly from that signal. But there's an additional feature that I alluded to, which is, or additional two features that I alluded to for PacBio sequencing, which are the pulse width and the interpulse distance. Pulse width, I've labeled on this plot that I've taken from a poster presented by, I think, Chris Saunders at PacBio. Pulse width is just the duration that you see a signal from one of these nucleotides. And interpulse distance is the time until the next the incorporation of the next base. One of the interesting features that's annotated on Chris's image is the adenosine base in the middle of this plot versus the two A's at the end of the plot. And you can see just the amount of time it takes to move from incorporating that first A to the T is much larger than it is for the other two A's. These three features, the fluorescence intensity, the pulse width, interpulse distance, can be used to create a set of features that's fed into a neural network that is trained to call methylation signal. And so we see without bisulfite treatment or oxidative bisulfite treatment, we can see the methylation status of certain nucleotides without having to do an additional experiment. I'll let me skip that to this. Nanopore sequencing is very similar. Rather than the measuring the pulse width and interpulse distance, though, they could do it directly from the electrical signal, right? If you think of each nucleotide as a resistor going through this potential difference between between one end of the pore and the other, each nucleotide, methylated or unmethylated, has a characteristic signal. Now, sometimes that signal is not going to be so different from the unmethylated signal that the base caller can tell, but for 5MC and 6MA, it is. You can see just the third position in the current versus position in reference plot, a difference between a base and a modified base that you can train the base caller to identify. So the nice thing is that long-read data contains 2 omes in one, right? You have the primary genome sequence for your sample, but you also have a 5MC readout for your sample without having to do any additional library treatment. There are comparisons between Oxford Nanopore, PacBio methylation, and either bisulfite treatment or oxidative bisulfite treatment. There are a lot of different versions of this, but they all sort of come to the same conclusion, which is there is very high correspondence between the Illumina bisulfite treated data sets and the PacBio and Oxford Nanopore. There is a gap, but that gap is due to PacBio and Nanopore being able to access more parts of the genome than the Illumina sequencing can, right? Because remember, for bisulfite treatment, you're basically reducing the complexity of the sequence by turning all unmethylated cytosines into uracil and then through PCR amplification into thymine. And for oxidative bisulfite sequencing, you're converting both unmethylated cytosine and hydroxymethylated cytosine into thymine bases. You only have 150 bases, so if you reduce the sequence complexity of that read, it's harder to map those reads to where they're supposed to go. With the methylation readout in the long-read data, you're still benefiting from the full length of the long-read data without having to convert any of your bases to another base in order to read out the signal. So there's no loss in the actual sequence accuracy, and on top of that, you get a layer of information that you would have to go out of your way to get for Illumina sequencing. Oh, sorry, go ahead.
About cost.
Yeah, I guess that's right. I mean, I haven't, in my day job, I try very hard not to think about sequencing costs because they change so much and it's not my department. It's not my, it's not my issue to deal with. But you're right, like you would need to do 2 experiments for Illumina sequencing versus the 1 experiment that you need to do here for PacBio sequencing and for Nanopore sequencing. That does, basically double the cost of Illumina sequencing. I don't know many people who necessarily do both of those experiments. The way I think about it is it's actually kind of in the other direction. I think because of the additional treatment, because you require more DNA, you require more time, prepping samples, increasing the cost. I actually think that most short-read studies that I've seen probably aren't generating the methylation data unless they absolutely need it, unless they really have a hypothesis. Here, you get it for free. So all of a sudden, there's a whole bunch of analysis that you can do that you might not have considered before. Like, for example, at repeat expansions, can I see hypermethylation in the region of the repeat expansion? I might not have considered to look for that before, but now I can see it without having to do any extra work. One of the questions I'm often asked about this is, how do I get my methylation data off of the instrument? Does it come in a bed file? And the answer is, you don't have to do anything. It comes automatically with the sequence production process. I put a little asterisk. I put a little asterisk on that because Nanopore, this is true for Nanopore as well, but Nanopore gives you a lot more control as to what settings you use in base calling. So it's worth checking that you've enabled this setting, or your sequencing provider has enabled this setting for Nanopore data. For PacBio data, it's on by default. It doesn't come as a separate file. It comes embedded in the BAM file that you get. So if you get the BAM files that you're doing SNP and indel and structural variant calling on, that BAM file has the methylation data directly encoded in it. I won't go into details on exactly how that encoding works. The important thing to know is, two things. One, it's embedded in the file. Two, most of the tools that you will be using are adapting themselves to handle this signal natively. IGV has already been updated to show methylation information. All one needs to do is right-click and ask to show the methylation signal as opposed to the primary base signal. If you're writing new software, then I'll tell you that the MM and ML tag, which respectively denote base modifications, and the probability of those base modifications is a standard part of the BAM spec. HCS-JDK and HCS-lib have been updated to read and write those tags. So there's a lot of the bookkeeping that we need to do in order to read that signal out appropriately along the length of the read sequence, but a whole bunch of software now exists for you to do that pretty easily. Okay, with the time left, there's one other thing that I wanted to talk about. I think this entire time I've been talking about interesting consequences of single molecule sequencing and what kind of readouts we can get that we couldn't get with Illumina sequencing before. There's one more that I think is really interesting called adaptive sampling. So one of the problems that we have with that my group has been approached with several times is a colleague who has a locus in the genome or a few loci in the genome that they think something interesting is happening at that locus. Either it's a structural variant or it's a de novo mutation, but they can't get enough of a clear picture of that locus from short read sequencing. At the same time, it seems like a waste to do long-read sequencing on the entire genome just to see a handful of loci. Now, there's stuff you can do with PacBio sequencing around targeted coverage. PacBio has a number of targeted sequencing kits that target repeat expansions or clinically relevant medical genes or the HLA. But if you just have another locus in the genome that you're going after, you may have to do PCR over that locus or some other strategy in order to capture what you want. Versus nanopore, where the targeted sequencing doesn't necessarily require any new library prep whatsoever. Instead, because nanopore sequencing is based on a pore rather than a polymerase, you can reverse the action of the pore. And that's a really interesting prospect if you're trying to target the genome and only generate reads around where you're looking. So basically, the way this works is you start sequencing some strand of DNA. The base calling readout is in real time. So you have a chance, as the read is translocating through the pore, to read what the first 200 bases of that read say. And if those 200 bases are not originating from some part of the genome that you are interested in, you can reverse voltage on the pore, which essentially unwinds that fragment of DNA. Then you reverse it again, and you allow that pore to accept new reads. Right? If you think about each pore as having a sort of lifetime for the total number of bases it can read, well, you're not reading the entire read. You're not using up all of the capability of that pore just yet. So you reject the read, you wait until you see a new read that has 200 bases of leader sequence that you care about. There are a number of different groups that have worked on this. I'm showing the papers from Alexander Payne and Matt Luce. I know Mike Schatz's lab has worked on this. A number of different labs have worked with schemes with this. Basically, Nanopore gave an API for real-time control of the instrument, and let researchers play with what they would do with it. I like Alexander's papers here because they highlight a few features that I think are really interesting. First is if you have control over individual reads, you can do kind of clever things like target repeat expansions, target copy number variants, target structural variants. You can also barcode all of your reads and only accept reads that have a certain barcode. Let's say I have 8 samples that I'm trying to get data on, but I want an even coverage distribution. All right. So once I've seen enough data on barcode 1, I don't need it anymore. So I could just have the software reject that barcode for the remainder of the sequencing experiment and prioritize the ports towards other reads of interest. I think there are three things that I see people do with this capability I think are really interesting. So that dynamically switching on and off barcodes depending on the coverage achieved, targeting a specific barcode. So if we've seen enough coverage on, say, a variant of interest, we don't need any more data from that barcode, and we can again prioritize other ones, and we can just generate more coverage over some locus of interest. Right? Think if you're doing a genome assembly and you need to close gaps in that assembly, you could just generate targeted data over those gaps rather than having to do yet a full other sequencing experiment. This capability is best when one is sequencing around 1% of the genome or less. So we can see from, I think this is also from Alex's work, so that we can see from the, from this coverage that around 1% gives us a 14 or 15-fold improvement in the overall coverage that we would have gotten at that locus if we had done no targeting at all. We had just done whole genome sequencing. This works for pretty much all of the forms of nanopore sequencing. So from the handheld instruments all the way to the production instrument, this sort of targeting is possible. And I'll end with what I think is one of the most interesting applications of this is going after target repeat expansions. This particular repeat expansion is on the Huntington's locus. Similar efforts on FMR1 and a number of other repeat expansions that would be not accessible with short read sequencing. I think this is a really lovely approach. And only possible because the nanopore instrument is the one instrument that is not polymerase based. Okay, I want to leave time for questions, so I'm going to stop there. But before I do, let me just thank the tremendous number of people I work with. Again, this list is out of date, but the people who I work with who have contributed to this presentation in some way or another, I'm enormously grateful for their effort and guidance, and I appreciate all of you for listening to me. Thank you very much.
We have a question online. They asked, how many targeted sequences can you do in the targeted study?
It's a good question. I mean, I don't, on the one hand, I don't think there's necessarily a practical limit. Nanopore does provide barcodes in, a few different configurations. I think, 4, 8, 16, up to perhaps 90. I can't remember what their limit is. But more practically speaking, there are two things to know. One, you can estimate the yield of a flow cell to be. I generally think around 90 gigabases for a PromethION flow cell. So 90 gigabases divided by the number of barcodes. But it turns out it's not that simple. There is some penalty for using barcodes. There's some yield penalty that we've observed for using barcodes. So we tend to think it's around 2 to 3 barcodes. Now there'll be back and forth. I mean, there are constant improvements in these platforms. So it's something that we need to check in over and over again. And every time Nanopore updates one of their pores, they also need to update one of their barcoding kits. So there's a bit of a lag between upgrading the pore and upgrading the barcodes. But that's generally how we think about it today. Is around 2 to 3 barcodes gets you like 30x or higher coverage over targets as long as they're less than 1% of the genome.
Aaron, thank you for the talk. And I have at least one question. Are there data sets now that are emerging that are combined, like with this combined methylation and sequencing data?
Yes, there are. So wait, let me just clarify. Are you asking, are there publicly available data sets that have both primary base signal and methylation signal? Or are you asking, are people combining PacBio data and Oxford Nanopore data? Because I'd be happy to answer both of those questions.
The first one. So like when you were talking about being able to kind of read out both from the same.
Yes, there are. So publicly available data sets are available on PacBio's website. Every time they've upgraded the instrument, they've released they released a number of things. They release usually HD002, but they've also released some tumor normal pairs. I think there are a couple of colorectal cancer pairs on their website. They have a few microbial genomes as well, which for 5-MC methylation might not be as interesting. But there are a bunch. There are a bunch there. The second place to look is the Human Pangenome Reference Consortium, and I kind of treat them together, but they're separate things. HPRC and Human Genome Structural Variant Consortium. Now, I will say the early data that they generated, I think they stripped out the methylation signal. So you should know that the ability to call methylation on board the instrument is fairly new, maybe only in the last few years. So the initial data that was generated by HPRC didn't contain that data. Newer data coming from HPRC, I think, should contain that data. And all of that is publicly available. It's 1,000 genome samples, so all of that is available. And then third, so it's not publicly available. Well, yeah, it's not publicly available yet, but it will be in the next, probably within the next month or so. What's called the V8 release of All of Us will now contain a couple thousand long-read samples. We had released about 1,000 long-read samples in V7. They didn't, for the same reason I just indicated, they didn't have methylation signal. The new data, we've been very paranoid to make sure they all have methylation signal. That's the PacBio data and Nanopore data. That's around, I can't remember the exact number, but it's somewhere between 1,000 and 2,000 genomes from a number of individuals from diverse ancestry. And that will be available on Researcher Workbench for anyone who wants to access them. No problem.
All right, I think we have time for one more online. Are there mature base callers for other cytosine derivatives, e.g. 5-HMC or 5FC?
Yes, there are. I actually don't have a comprehensive list in my head as to everything that the base caller supports. I think it's 5MC, 5-HMC, and 6MA, but 6MA calling I think is usually for RNA. I assume it's been updated for, I think it's probably capable for DNA as well. I'm just a little bit less familiar with that literature to be able to say for certain. And it can also be trained to detect different modified bases if you have such a training set. Nanopore releases their base caller as open source, and they provide documentation on how to train it for new base modifications, which is a pretty exciting thing to consider for a number of RNA sequencing applications and other synthetic biology applications.
Great. And then there's a few more questions online, but we're out of time. So if anyone can email Kiran with additional questions, I'm sure he'd be happy to answer.
Absolutely.
Thank you. See you after the new year.